Good afternoon, everyone. I am starting this live stream. I'm waiting for Dmiter to come in. Okay, he's already online. Hello, Dmiter, can you hear me?
Yes, I can.
Just a moment.
Okay, I'm here. I'm going to make a coffee and I'll be with you. So.
Hello?
So I'll go make my coffee and I'll be with you. So are we expecting someone else?
Just a moment, please.
Yeah. So it's look like that Mike is kind of exhausted. He explained. Very tired, probably. He was on a live chat this morning. I saw it.
Yeah, I didn't follow it. He started to talk about how he doesn't believe in appearances of people who testify.
Hello? Now
Yeah,
it's
just a
better.
moment, just a moment. Yeah, so I was saying that I was just following him shortly, and he was talking about, he was putting up a video of James Tour about his testimony that Christ appeared to him, and so he was talking about how he doesn't believe in these kind of appearances and so forth. So I'm a little bit, how can I say, bored about the expression of incredulity, because we have testimonies all over the world from so many people who have made supernatural experiences. So um, I'm not very interested to hear about people who do not believe in these things. So I didn't follow his chat this morning. So I don't know what else they have been talked about.
I mean,
I mean,
I'm
I
not,
would wonder
yeah.
how do you, beside you being a scientist doing abiogenesis research, just like maybe a side note, I wonder, I don't know you, I don't know you well. What is your position, your ontological position in regards of existence in a holistic sense, let's say?
Well, first, a lots of our understanding and our feelings could be, I mean, so whatever we see, whatever we feel, whatever we watched, whatever we experienced, uh, definitely it's not our complete picture. It doesn't matter how powerful, how compelling could be our experience. I mean, it could be very trustful, could be extremely important for us. Uh, I can speculate that one of the reasons that people so much likes to believe in something, it doesn't matter what it is, it's just for the reason that they feel better this way. They feel good about it. And
Well,
that's
I can give you an example. I have not made very often.
so it's totally different question what actually is the reality and what, since we experience something, is it possible this experience to be because of some intelligent source or just because the source is intelligent? Meaning our own brain, it's just feel the situation, just feel the reality in this way. But I'm completely in respect of the people feelings. I cannot say there is nothing like, I mean, the experience of your own, how to say, deep inside personal feelings are something very important.
well, we are
Uh,
not talking about feelings, Dimitar. In case of James Tour, in the video which Mike showed this morning, James Tour, he gave a
I didn't,
testimony of.
I was just witness a little bit from this. I was kind of busy, so I don't know exactly what everything was this morning.
I just
Uh,
want to focus in regards of the small section which I saw, where he showed a testimony of James Tour, where he talked that he had an appearance of Jesus. So he didn't exactly just testify a feeling, but actually a supernatural event where he had an appearance of Jesus, a
well,
miracle, let's say.
this is experience. I can say for sure this is some sort of experience. There is no doubt about that. If people see Jesus, uh, for example, because he's coming from uh, background of Christianity, in the same way, for example, the people that come from background of Hinduism, for example, they can see Brahma or Vishnu, and
So,
they can
so
see
could it,
this way.
yeah,
Uh,
so could it be, Dimitar, that there is actually a spiritual world because the Bible tells us also about demons, about Satan, about angels, fallen angels, and I want to give you an example about an experience which I made personally, which I can testify. And it was in our state. There is a Brazilian a businessman, and he converted to Christianity, to Jesus, and he bought a trailer, a bus, and he stopped to do business. He is a very rich man, and he started to tour in every of the 76 small municipalities of our state, and there he made evangelism and proclaiming Jesus and called people to come to believe in Christ. And I was invited by a pastor, a friend of mine, to join one of these evangelism campaigns, and I went with my friend. And besides the bus, there was a small tent. And when someone had an issue that he wanted to convert himself or so, then they invited these people to come to that tent on the side of the bus and then just talk to the ministers and pastors and so forth, and explain whatever problem they had. And I was present there, and the lady came in, and as soon as she came in, that was in a municipality where there were many African Umbanda cults performed, and this woman, she was part of one of these cults. And as soon as she came in, she started to be possessed by a demon, and he expressed himself. She started to scream, and the pastor, he interviewed him, and that woman, she was for about 10 or 15 minutes possessed, and then this pastor, he ordered the demon to leave that woman in the name of Jesus Christ. And immediately when he invoked the name of Jesus, that woman, she fell on the floor. She stayed silent there, like dismayed, for about 5 or 10 minutes or so. And when she wake up, she was like not even knowing where she was. And then she testified that she was consecrated to a demon, to that cult, to that Umbanda cult, when she was 7 years old, through blood. And since then, she was frequently possessed, that demon destroyed her life. And at that moment, she said, the first time in my whole adult life, I feel free, and I feel that this demon has left me, and he is not controlling me anymore. So what I think is interesting in regards of this event, which I personally experienced, is that that demon didn't leave her in the name of John or Dmiter or Otangelo or any other name. It was clearly invoked the name of Jesus Christ. And I am not talking about whatever kind of experience other people had. I am talking about a first-hand experience which I presenced, actually. So I say that as a first-hand testimony of what I saw. And that experience was very parallel to many experiences that we can see. If you make a search on YouTube or so, you will see many testimonies of people all over the world which will testify exactly the same kind of experience. And if you check about Umbanda, for example, they make cults where people are possessed by spirits, and they are clearly changing the physiognomy of the person, and they start to talk in different voices and so forth. So this is not a unique phenomenon. This is a phenomenon which is very widespread and which is also very old. Even in the Gospels of Jesus, we have things about men and women which were possessed and which were freed through Jesus Christ. So, of course, my view, and whoever wants to say, well, this is something, a fantasy or so in this sense. But when we have a certain number of testimonies which have the same kind of experience, and we know this experience all over the world, then it is, in my understanding, very difficult to say, oh, this is just bullshit, this is just fantasy, this is just feelings, or whatever kind of objection that an unbeliever comes up with. So I am sure that I'm not the first to talk to you about this kind of things. You know that this exists. How would you give an interpretation to that?
Well, I have respect to the feeling of people, but this is your explanation. Everyone's explanation comes for the background that he have and she have. That's what actually, it doesn't matter who you are. Everything that you explain around you is based on what you have as a background, as a information behind you. So, and that's very simple to understand that. Uh, Explanation is that is your explanation, and it makes sense perfect to you because you know it's written, it's there, and many cases in the past and so on, and that's your explanation. So what I'm trying to say is it doesn't matter what is your explanation is in most of the cases it's not complete. It's not complete. I do not know everything. You do not know everything. I have my own background of education and information and something that it's embedded in my brain based on my childhood experience. And after that, based on my reading and so on. And based on my feeling as well. Just 1 second. Sorry about that. We always have to pay attention of our wife's messages and calling. so, okay. So my point was, first of all, we have to acknowledge that we are not in position always to say something which is firmly to say, yes, that's it, yes, that's nothing, because we are not absolutely knowledgeable uh, creatures.
Yes, I agree with that, and that's what I
So
always say.
that
We
so
have no access to ultimate reality, to what is behind the curtain of the universe. We
yes.
don't know if there is or if there is not a
So,
God. So we deal just with the evidence. And
let
what I told you is, as I said, first-hand experience. I'm not
no
talking
no.
about what I heard from others. So we have that evidence that in the name of Jesus Christ, people they are freed from demonic possession. Okay, demonic we can of course that's questionable, but people are getting possessed. That is reality. This is not just made up. I have seen that not just one time, many times. That is reality, and that these people are freed from this kind of possession in the name of Jesus Christ. When the name of Jesus Christ is invoked, this is a fact. This is something that we observe. So the question is, what do we do with that evidence? How do we give an interpretation to these things?
So let me finish whatever I tried to say.
Okay. Mhm.
So for you, as I mentioned, for you this is evidence, and your explanation is your explanation, which is coming based on everything that you have as a knowledge, as understanding, and that's fine. Everybody should explain the things according to what you cannot explain something that you have no even slight background on over it. Now, is that mean, even that it's your explanation, even that this is your explanation, is that mean that the world, I mean, okay, so there's many questions here, but let's forget about most of the questions and ask one question. Is that mean, even that this is your explanation, is that mean that the world, the phenomenons that is happening around you have to be irrational? So
Well, the question is what I told you is the evidence. That's what we have, what I have observed to firsthand, and we
you
need
see,
to try
okay,
to give an adequate explanation of the phenomena which we are talking about. So my upon my understanding and my worldview, not just based on that phenomena individually, but taking all the evidence together, which is the Bible, the Gospels, the New Testament, what we read there, experiences from other people, my personal experience, and that joined together with the observation of the natural world and so forth. That gives me a support to believe that there is a God and that the theistic worldview is true. That is my point, my epistemological, ontological viewpoint. How would you explain that phenomena in particular, that demons are cast out not in the name of any person, but in the name specifically of Jesus Christ?
it is so I can I'm not in position to go right now to explain all of that. However, this is could be explained in a psychological way. So everybody in his particular background and environment of specific beliefs, of specific understanding, always. So let me say this way: imagine a person in a remote island. Let me say, for example, Eastern islands, and the people who grew there, they have never been they've never been exposed to Western or Eastern culture. They have some their own mythology, they have their own myths and legends and so on. Imagine this person is possessed by a demon, and suddenly someone come from abroad and lay hands over him and say, in the name of Jesus, this demon gotta go away. The person by itself doesn't mean anything. What does it mean, even the word Jesus? He's never been exposed. He even didn't know anything. Like, for example, I can say something to you in the name of Junja. Go ahead. Something. You don't know what is Junja. I don't know. I just made up this up.
Yeah, sure.
But my point is, you're predisposed in advance to certain types of certain types of beliefs. You're predisposed because you grew up in this particular environment. So now let's see. I'm not going to start debating this because there's no point. This is just some sort of my explanation that could be, without going deep into the
yeah. We will change our topic afterwards.
And again, people from totally different environment, they have absolutely the same experience, just the names are different.
But that's what I'm saying. Person which is possessed, Dmiter, that person has no conscience because the demon is possessing the body.
That is your explanation. Again, this is your explanation that he or she, so she or he is possessed. This is your explanation. And it's also your explanation. It's also your explanation that he or she is free of. So this is two relatively controversial explanations. What does it mean possessed? What does it mean free of? Yes, in one case he looks wonderful, in another case he looks terrible. I agree with that. But this is your explanation based on your background and based on your understanding. And based on whatever has been, yeah, exactly. You're observing, but what you're observing what is observing from outside. You're observing something that you do not observe actual feeling of the person. You do not observe.
Of course not. We don't know what is happening inside the person, but what we observe is that person expresses another spirit, which actually even changes its voice, its intonation, and when the pastor asks the name of that spirit, he actually even answers the name of the spirit. The person that is normally that person is not that spirit.
I understand. You saw.
That's what we see. And we see that the power of the name of Jesus Christ actually is able to cast out that demon, and that person returns to have a normal life. And in particular, that lady, which I experienced, which I saw with my eyes, when she woke up, she said, I am finally free after an entire life being bound to that spirit. Since I was consecrated when I was 7 years old in a ceremony by blood to that demon, I am bound to that demon. And at that moment, when she woke up, she said, I feel peace. And the first time in my adult life, I feel that I'm not bound anymore to that demon. And this is not something that has to do with my religion or so. It has to do with evidence. It is what we see happening in real life.
No, this is not evidence. This is observation.
Yeah, evidence, observation.
You're mixing two things: observation and evidence. I mean, observation is observation, and I agree. But to say that this is evidence, it's not evidence. It is observation.
If you give the meaning of evidence as the explanation, then I agree with
That
you.
you saw, I understand. Because you saw, you think it's evidence. No, because you saw doesn't mean it's evidence. So, meaning you saw something which is impressed, but also there is a possible explanations different than whatever you think it is.
Yeah,
I'm not going to argue. I'm not going to argue at the moment. This is not the topic I want to enter into.
Yeah.
But just one question. Okay, let's assume you believe in that. You're thinking that it's true. Let's assume all of that it's true. Although I'm saying,
Worldview.
yeah. So let me assume that is that mean the world is irrational and it's not logical?
No.
No. So once something is, so you have totally different from, for example, when we say proof based on a science, that's a different. So because the scientific proof is go through specific experiment, and the specific experiment have a hypothesis, and this hypothesis is just tested by experiment, and you have predictable outcome based on that, and this is proof. And also something else you have to know, what the science work with. The science does not work with the truth. We never intend to work with the truth. The science, it's not about a discovery of the truth. The science, it's about facts. It works with the fact. That's what the science is. So you have fact, facts, facts. It's not about the truth. Truth is something, could be personal. Truth is something, could be whatever you want to call it, more, or some assembly of many facts. But we do not work with the truth. We work with the facts as it is. That's why you have fact, experiment, hypothesis, and again, fact. So it's a process of self-exclusion. In that case, you go in some sort of objective direction. It's not subjective direction, it's objective. For example, I can always say that this particular bottle is a red bottle. But in fact, when you scan it and see the reflection of the light and see the number of this reflection, you see that it's blue. See, it is kind of different. The scientific process is more objective process, and it doesn't go and doesn't do something like claiming of something huge, enormous. So from the perspective of cosmology, there is suggestions how the actual universe is built up, what the elements are of the universe. But again, is this his entire picture? No.
I said we will never have the entire
picture. It's not entire picture.
But you cannot see the universe.
It doesn't matter.
A complete picture.
Okay, but there is one thing you should understand and I should understand. It doesn't matter what is our level of understanding to entire picture. It could be very small or could be bigger. It's still you have a process of logical proof and disproof, of logical, experimental, and factology things which are logical. They are not irrational. They must be rational. This world is rational based on a law. Now, because we do not know everything. There is no person in the entire world here that can say, okay, I know absolutely everything everywhere. That's not such a case. Again, I'm saying the same thing over and over again. The science does not work with the truth. It works with facts. And based on the fact, we can draw conclusion a little bit further. Based on the facts, we can draw and extrapolate the picture which goes to more real picture than it is. A typical case. Back in the medieval times, solar-centric system versus geocentric system. So first you have observation that indeed the planet Earth is the center of everything. But once you start to ask the questions, hypothesize the questions, and you go for observation, you see another facts which draw to another conclusion and draw another hypothesis, which maybe the observation fits much better if we put the sun on the center. In that case, if we hypothesize that the sun is in the center, therefore we have to observe that, and that. And let's observe it. And you observing can see, oh, all the facts fit this hypothesis. Therefore, solar-centric system is more accurate to represent the reality than the Earth-centric system. But, in fact, even sun is not the center of everything. We know now that the sun is rotating over the center of the galaxy. And we know now that there is billions of other galaxies. We know everything in the last few years. I mean, everything what we know, this modern thing from the last few years.
Yeah, it is obvious. The more that science advances, the more we will change also our understanding of reality and change science.
Yeah, so in that case, do we have more objective view to the universe, to how the nature works, in compare with what we knew 100 years ago or 1,000 years? It's more. And this is based on the same system of science. Now, that means the explanations sometimes are changing for the same phenomena at the same way.
Yes.
Because you know more now.
Yes, yes. I want to go back to what we were talking and actually even finishing yesterday afternoon, Dmiter, where you explained your explanation or hypothesis of proto-translation with the amino acid bound to an RNA. Okay, and I was thinking about this this morning, and of course, the more theoretically that development advances, at some point you come to a tRNA, which then is the adapter molecule, which is used by modern cells to perform the translation.
Mm-hmm.
Okay, now what I think basically kills your hypothesis is this: the tRNA is a very complex molecule which has to be made by other molecules and proteins, which it has to go through a very precise biosynthesis process, which is very complex, and it is a multi-step process where many steps are involved until you come to a mature tRNA. Correct?
So, let me see, do I understand you? You said that the initial proto-tRNA will become fully developed, mature tRNA on many steps and process involved. Is that what you want to say?
I put it here. I have an article on that. Okay. Transfer RNA and its biogenesis best explained through design. So I am describing here is the tRNA molecule, okay, and in to give an example, in tRNA maturation in Homo sapiens, following enzymatic complexes are involved in the process. Here are the proteins and then the enzymatic complexes. We have 5 different steps mentioned here to make a mature tRNA. Okay? So, of course, if you need tRNAs prebiotically in order to start the entire translation process, the tRNA has to be also processed and made prebiotically. But we have an entire process based on enzymes which are involved in the maturation of the tRNA, but that process has no function at all, neither the process nor the proteins, unless the future goal to have a tRNA with a specific function is known beforehand. And tRNAs by themselves, they have no function unless they work in the joint venture together with the other proteins and enzymes like the ribosome, the amino acid tRNA synthetase, the messenger RNA. This complex works all together. So a mature tRNA by itself has no function. And so it would make no sense for all these proteins and that manufacturing process to be there to have a mature and functional tRNA without the other proteins. So this is like, I can give you another example. In photosynthesis, this is equivalent to a photovoltaic system where the first molecule is the chlorophyll. This is the light capturing molecule which gets the photon. But if you have only the chlorophyll and you do not have the antenna complex and all the other proteins used in photosynthesis, then chlorophyll by itself has no use, has no function. Now, chlorophyll also requires a complex biosynthesis process of 17 different proteins which are involved in the maturation of chlorophyll. But if you have only the chlorophyll and not the other proteins, chlorophyll has no function. That is a parallel to tRNA. If you have tRNA, but you do not have the other proteins involved in translation, tRNAs by themselves, they have no function. So why forsake would abiotic biosynthesis pathway, which is complex using a multi-step process involving many different enzymes, producing a tRNA which only has function in a joint venture together with all the other proteins in the translation process?
Just give me 1 second. I'll be back and I'll explain to you. Yeah, I understand the question. So just give me 1 second.
Hey,
Hi.
you guys were talking about tRNA. I didn't quite hear the question though.
My question was that it requires a complex biosynthesis process to make tRNAs, and that process would have to be also extended prebiotically in order to have the entire translation process. So these proteins, enzymes which are involved in the tRNA maturation, they would have to emerge prebiotically together with tRNA. But tRNA by itself alone, not involved in the translation process in a joint venture with the other proteins, which would have no function. So upon my understanding, translation is an irreducible complex process. You can only have translation if all the proteins and enzymes and the ribosome and so forth are in place and working together.
So the issue is that there's a very small region of tRNA that's actually responsible for its activity, and that activity is based upon the secondary and tertiary structure of RNA. And those structures form without the assistance of many proteins.
That is not my point. My point is that you require a complex specific enzymes in the maturation process of
Right, but
tRNA.
my point is that in the beginning of all this, you don't need those proteins to have the most basic functional unit for activity. You don't actually require those proteins. What you require is the portion of the ribosome that has the activity.
Well, the tRNA is there, and the tRNAs, they have to be made somehow. And the
Right,
whole
they're formed
translation process has to be there in order for the whole chain of protein manufacturing can happen. And we know that proteins are life essential, and so is transcription and translation. And in the translation process, tRNAs are required.
Right, but so the majority of tRNA, the proteins that are found in it, are found in order to stabilize the structure of the RNA strand.
They are found where? Sorry, I didn't understand.
Most proteins that are bound with tRNA are bound in order to stabilize it. But it's the series of nucleotides. But it's the series of nucleotides within that string of RNA that actually has the catalytic activity. And that structure that it adopts forms naturally.
Well, the tRNA is synthesized by a long process of maturation by different enzymes, correct?
Are you talking about the RNA strand itself?
No, I'm talking about the tRNA molecule.
The proteins would be, yes.
Okay, so we need a complex manufacturing process to make tRNAs, okay?
Okay, but do we need tRNA as it exists in eukaryotic life? Or do we just need tRNA that has catalytic activity? For transferring, for creating peptidation bonds?
Well, my starting point is the tRNAs as they are.
Okay, why would it have to be as it is?
Well, I don't know if eventually some kind of simpler or different tRNAs would be the same function, but we know that they at least need the two binding points in order to permit the translation and the decipher and the assignment of the anticodons to the amino acids. So at least that had to be there.
Right, except there's research that already shows how, or that postulates how tRNAs have evolved. And
well, first of all, the origin of tRNAs, in my understanding, is not an evolution problem, but an abiogenesis problem, because the translation process had to be there when life started.
Yeah, sure.
Okay, so the question about the origin of tRNAs is not an evolution problem, but an abiogenesis problem.
No. I mean, okay. I just wonder where to start, basically, to explain all of that. I mean, the model that that is embedded. So based on what you showed me, it's embedded completely wrong model for the function of the appearance of tRNA. Yes, in the modern situation, you have a process of transcription and after that maturation of tRNA. You have involved many enzymes to produce the modern tRNA. And there is a genes for every single tRNA, which is transcribed. And after that, it is maturated based on several factors, including one ribozyme. It's called ribozyme P1, which is ribozyme is enzyme from entirely by RNA.
Mm-hmm.
And this is correct. And there is additional enzyme which is after maturation, it's adding CAA on the end of on the triprime end of every tRNA. This is the modern view on it, what is happening at the very full developed form. However, that doesn't mean the tRNA function as tRNA always in this particular way. So you have fitted over fitted. You can have a very primitive translation with very small RNA, which possess the function of tRNA. Because in fact,
Just give me a minute. My dog is loud here, and I'm distracted. Just a minute. I have to close the door.
yeah,
Okay.
yeah,
So
go. That's what the dogs are selected for, to bark. Hi, Mutual.
Hey, how's it going?
Okay, okay. Enjoying?
Oh, it's
So,
always fun.
yeah. Yeah, I can need your support. Otangelo, yeah. So sometimes you can explain it better than me. Sometimes I can explain it better. So we'll see how it goes.
Well, at the very least, I try. It's not always the simplest thing in the world, but
Yeah. So if you go to, I mean, I might repeat myself millions of times, but uh, you know my two papers, right? In
yeah, I do.
2019 and 2020. So the paper which is 2020, the last paper, it describes the origination of genetic code.
Yeah,
So, and this is very important because that is the key for origin of life. So,
okay. Sorry.
Otangelo,
Yeah. Go ahead.
I wrote here Zachary Burton. Here you see the name Burton,
yeah, I
Zachary.
do.
So you can go in PubMed and you can see tRNA and Zachary Burton or just Burton Z, and you'll find one of the best hypotheses for how the tRNA actually evolved in terms of uh, the actual repeats of nucleotides that you have. So you can explain the origin of tRNA very relatively simply by amplification of some short oligonucleotides. And you can see even how it fits very well the entire. So you can think about tRNA as a duplex of something that is just duplicated and became two and folded. Now, the modern tRNA works with the modern ribosome and fits very well. However, if you have a primitive tRNA, you you will not work with the modern ribosome. So you have to have primitive ribosome as well.
Right.
The entire model that we are working with, the scientists working with, is you have a primitive RNA and primitive ribosome and primitive system which start to co-evolve. You have a system of co-evolution. One and another is changing each other and gain more and more specificity and more uh, became more functional, more efficient, with more high fidelity. Uh,
So Dimitri, let me please interrupt you. I want to ask you. Let us suppose that tryptophan would have been the last amino acid added into the protein set of amino acids. Okay, so let us suppose that there would have been a ribosome and tRNA, 19 tRNAs, and the whole instruction in the messenger RNA without instructing to make tryptophan. So how do you imagine that there would be an expansion or an evolution of an alphabet with 19 amino acids to the addition of one more tryptophan? First of all, to make tryptophan, you know how complex that is in a modern cell. It is extremely complex. It requires the entire metabolic pathways like the Shikimaki pathway and so forth to make tryptophan. But let's that put out for an instant. So let's suppose suddenly from a system of 19 amino acids we go to 20 and tryptophan is added. We need to have a new tRNA which we can make the translation. Then we need the amino acid tRNA synthetases which can recognize tRNA, and we need probably also to change the ribosome in order to expand to have the addition of that amino acid, and we need to change the genetic code. So we have at least 4 different things that have to be changed. And how do you imagine that this could have happened prebiotically? Because as I understand, at the last universal common ancestor, at least all the papers that I have read, the amino acid alphabet and translation, the whole system of protein synthesis was there. So I think this is an enormous problem. And actually, I don't know any science paper that comes up with a reasonable explanation of how that could have evolved in a stepwise fashion.
Well, there is a scientist which explained very well how it happened. There is a very reasonable hypothesis for that, and you do not need everything to be changed at once in order to have another amino acid to be assigned. So assignment, how go the assignment for new amino acid? It's all started with similar pairs. So if you see, for example, a codon, so the existing codon could mistakenly be I mean, not mistakenly, but let me say in non-perfect situation, you always can amino acylate with another amino acid, the certain tRNA.
Dmiter, let's start a little bit earlier. Explain to me why there was actually an assignment of 3 codons to an amino acid mindlessly, without purpose.
No. There is no mindlessly. There is a... Okay. First of all, what does it mean prebiotic and biotic? Prebiotic means everything that is before Darwinian evolution to happen. That means prebiotic. After, once you have Darwinian evolution, you have biotic. Now, mindlessly, it's not the correct terminology. The correct terminology is you can have a process where could happen by chance, but in fact, once it's happened by chance, it might happen also more suitable and more important for the survival of the system.
There was no survival at that point yet, because there was no life at that moment. There were just chemicals and molecules.
You're talking for deep. Okay, again, I'm repeating again. What does it mean there is no life at the moment?
To me, life is when self-replication starts. There are several things. The cell must be able to import raw materials. It must be able to adapt to the environment. It must be able to grow. It must be able to perform all the metabolic functions required to keep life. It must have the energy Uh, there are several things that are required in order to have a living cell, and that's the threshold from non-life to life.
Exactly. See, this is a wrong model. That's why you're confused all the time, because in fact, whatever you see, whatever you told me, it is already a whole life. But it's not only that. I claim by myself that, and the paper that I'm going to publish, the life is Darwinian, having a Darwinian evolution. It doesn't matter how complicated it is. You may have life only from two amino acids and five nucleotides of RNA. A complex of five nucleotides plus one short peptide could be called life. It's very simple, it's super simple. According to your understanding and your vision, life is something that should be and must be super complicated. But life, it's not about complication. It's not about how complex it is. It's about having Darwinian evolution. That's what the life is. Could be so simple that you cannot recognize it at all. For example, the system that have a primitive translation is already live. Primitive translation could be uh, 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 could be uh,
It could be as simple as a small
could be
peptide
assembled.
marker.
Yeah, could be assembled as a simple peptide and simple RNA. Absolutely, with a very small complex. That's what I'm trying to say. You're always working with complicated things, too complicated to happen. No, it's not like that. You have something so simple that you will not recognize it at all.
Okay. Paul Davies, he described life as reproduction,
As
metabolism,
information, I have about Paul Davis entire chapter in my new paper. He described the life as a information management. That's what he said about the
well,
life.
he made an article. I have put it here on the chat. I can also put it here privately to you guys. And he was giving an interview at the New York Times, and he described life as constituted having reproduction, metabolism, homeostasis, nutrition, complexity, organization, growth and development, information content, hardware software entanglement, permanence and change. That would be the functions required for a self-replicating and free living cell to be able to be alive.
So you don't read exactly what actually Paul Davis mean. I read his papers. This is all. So when you start to
But this is in
describe,
the article that he wrote. That's not me. That's not what
no, no,
I am saying.
I understand that this is not you. Paul Davis, and he even have YouTube lecture if you go there.
Yeah.
Paul Davis, and they published back 2015, I think, was a paper with Sarah Walker. Uh, and what you see here, reproduction, metabolism, everything, this is how you start the paper. You start the paper by saying, okay, the people usually describe life with metabolism, reproduction, having, surviving, and blah, 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 all of that stuff. And finally, by trying to find the specific features, what it is, if you continue read the Paul Davis, you will see something like life is information management. This is Paul Davis' conclusion after once he analyzed everything. And after that, uh, and I can tell you, this is not complete. It is correct, but it's not complete description of what the life is. And let me describe what the life is. So why it doesn't show? Okay, for some reason I'm not able to share the screen. Let me see.
I'm not visualizing it, so I cannot put it on the screen either.
Okay, I'm trying to share my screen now.
I try.
Oh, here we go. Okay. So, Paul Davies, his conclusion after reading of all of this paper and so on, you will see the Paul Davies conclusion is life is information management. That's what his conclusion is. My conclusion is slightly different. Life or Darwinian evolution, which is identical. There is no difference between Darwinian evolution and life. They are the same thing. Darwinian evolution or life is the same thing. It's a dynamically stable system where information determines its own supporting structure. Once you have this particular moment, you have life. Everything here, like replication, metabolism, everything is already embedded in this particular description. Dynamically stable system, meaning and supporting structure, meaning structure that could replicate information, but that could be very simple. It's nothing, that's what I'm trying to explain. There is nothing to do with the complicated things. It is simple. It's all about principle of interactions. Once you have information which execute some sort of structure, and this structure support the information or make it more stable, therefore you already have functional Darwinian evolution.
So this is instructional complex information which directs the making and sustaining of life. Would you agree with that?
Repeat again.
Information in that context, in biological context, is instructional, complex, codified information which directs and sustains the processes which are required to have self-replicating living cells.
No, it's not correct.
Okay, correct.
The term in cell. Okay, now I will stop a little bit sharing, and I will try to start a little bit back, to put back a little bit, in order to be very much more clear what is the situation. Immutable, are you with me?
Yeah, I'm with you.
Okay, good. Origin of life, the hypotheses of origin of life are many. And you know that. The most dominating hypothesis for origin of life is the RNA world hypothesis. However, there is additional different types of hypotheses. There is a metabolism first hypothesis. There is a peptide, a protein first hypothesis. There is a
replication, lipid world,
lipid world hypothesis, and many more. And
I've listed over 40 different attempts and approaches.
Exactly. So, what makes so let me just share one more thing, but I'll continue to talk to the moment when I'm looking for. Once I find the actual okay. So the different, okay, I will look, but so the different type of hypothesis, of course, they postulated somehow and how
the primitive or the translation happened to be and the replication happened to be. Now, you have to make a difference between self-replication as a molecular level, where you have RNA which is self-replicated itself, and self-replication in general, which is about the cell which replicates itself as another cell. So this is two absolutely different terminology, and both are self-replication. So when I agree that there is a self-replication in terms of cellular process as a whole, as a total, and of course we are self-replicating ourselves. But once it comes to the self-replication, talking about just a single polymer like a RNA that self-replicates itself, this is hypothesis which comes from RNA world hypothesis. By saying self-replication in RNA world meaning, exactly is that the RNA should self-replicate itself. Now, according to RNA peptide world, which I am following this is my hypothesis, our hypothesis. It's not only me that support this hypothesis. At the moment, probably around 50 scientists support RNA peptide world, not RNA world. According to RNA peptide world, there is no such a thing as self-replication in terms of self-replicating RNA. I'm not talking for the entire cell to be replicated. So see the difference. Also, according to us, RNA peptide world, RNA world is a wrong hypothesis and goes back to the history. However, unfortunately, by now, it's still the most dominated hypothesis, and many scientists continue to cite it and continue to say things like self-replication, and so on. So what is actually RNA world hypothesis? The RNA world hypothesis postulates that once you have a very short RNA, like several nucleotides RNA, they started to interact each other first. But at some moment, by those interactions, it happens that in the same time, when there is a short RNA, there is a completely natural, meaning non-enzymatic replication process. There is no enzymes there is nothing biological there, but there is a process of replication just in a chemical manners.
Has it been demonstrated that this is possible, Dmiter?
Yes, it is.
Yes, it has been.
I agree with that. So the experiments which shows Jacques Zostek, for example, and back to Orgel and 90s and probably early 20s, it shows clearly that yes, indeed, it is possible to have a replication entirely non-enzymatically, just adding the nucleotide without extra biological enzymes. And that could happen.
Yes, now, but has this been demonstrated in the laboratory?
Yes, it has been.
Yes,
or is it just theoretical? Because it surprises me, because Eugene Konin, for example, he says that it is not possible in his book Logic of Chains.
No. Eugene Konin, I know him. I read many papers from him. He is an RNA world scientist. He never said that it's not possible to have.
I can quote him if you want. I can quote where he says so.
There is a tricks here. There is a tricks. When you're talking for non-enzymatic replication, so due to environment, like a chemistry replication, And when you're talking about RNA replication, in most of the, in actually, it's a low, this is a low fidelity process, and also this process is specific only for G and C nucleotides.
He touches exactly that point.
It is almost impossible. It is almost impossible to make this particular chemical process with A and U nucleotides. And the reason is actually relatively simple, because as I explained much earlier, you have three hydrogen bonds. Yeah. So what actually who is saying this?
Konin. I'm citing Konin here.
Konin, yeah. So I'm saying what we know now, and yes, the experiments of the process of non-enzymatic replication are experiments and they are fact, but mostly for GC-rich RNA. So, in addition to this, next step, according to the RNA world hypothesis, next step will be, because you have maintenance, you need to have somehow a process of high fidelity non-enzymatic process.
That's exactly what he is saying here. I would like to read what he's saying in regards of this issue. Okay, Dmiter? The very origin of the first organisms presents at least an appearance of a paradox, because a certain minimum level of complexity is required to make self-replication possible at all. High-fidelity replication requires additional functionalities that need even more information to be encoded. The crucial question is how the Darwin-Eigen cycle could have started. How was the minimum complexity that is required to achieve the minimally acceptable replication fidelity attained? In even the simplest modern systems, such as RNA viruses, replication is catalyzed by complex protein polymerases.
Okay.
The replicate itself.
You continue. I'll come back. You continue I already know that. I already know. Just continue. I'll be back.
Okay.
Don't.
So the replicase itself is produced by translation of the respective mRNAs, which is mediated by the immensely complex ribosomal apparatus. Hence, the dramatic paradox of the origin of life is that to attain the minimum complexity required for a biological system to start on the Darwin-Eigen spiral, a system of far greater complexity appears to be required. How such a system could emerge is a puzzle that defeats conventional evolutionary thinking, all of which is about biological systems moving along the spiral. The solution is bound to be unusual. The origin of life, or to be more precise, the origin of the first replicator systems, and the origin of translation remains a huge enigma, and progress in solving these problems has been very modest. In the case of translation, nearly negligible.
If I can ask, when was this written?
In 2012. It's in his book, The Logic of Chance.
Okay, so it could be that the research I said isn't representative of the field necessarily. Although there have been several publications which show that if you take small strings of RNA and you place them and you have them in an environment that's relatively stable for them, so if you put them in a cold environment, for instance, and introduce a selection of the pressure for replication for them, you will have these small RNA strands that assemble themselves into ribozymes which have high fidelity in terms of their ability to preserve the identity of the strand.
Yeah, I am not aware of more recent investigations in regards of this issue. So it can be that I am outdated with that information.
Okay,
Yeah, I can see if I can find it.
Oh, sorry, Mutable. Can you continue? I don't want to disrupt you. Just finish.
Yeah, so what he was talking about is that Lee Cronin's quote was that you apparently need something more complex to allow for
Yes.
type of genetic information to preserve its identity.
Yeah.
Although
Yeah.
what I was mentioning is that
Okay, so let's finish. Let me say what is actually happening here.
Yeah.
Eugene Koonin, he is a RNA world scientist, and he even present one of his. He have a very good. I mean, his publication is his variant of how the transition from self-replication ribozyme goes to the translation. But his hypothesis is completely wrong to me. And anyway, but he is also Eugene Koonin. He asked absolutely legitimate questions, and he finds a difficult to explain how it's possible to come to the state of self-replication ribozyme. Why? Because in order to have self-replication, to develop self-replication ribozymes is relatively complicated, long RNA, and has been shown in vitro, in laboratory conditions, that you can generate artificially self-replication ribozyme, but this self-replication ribozyme always it's relatively long. One of the first self-replication ribozymes was about 140 something like that nucleotides, which is very long, and you have to have a specific sequence, and that cannot happen according to the statistic just by random assembly. Now, Jacques Zostak, for example, he said, okay, in order to come to that point, to appear to assemble naturally without any assistance self-replication ribozymes, you need a high fidelity of non-enzymatic replication first. Therefore, you have to figure it out how fidelity of self-replication occurred. How high fidelity replication occurred, non-enzymatic. That's why Jacques Zostak in his laboratory started to do some tricks, like, for example, not using RNA, but using RNA-like moleculars, which it happened that for some reason they have a much better fidelity and also incorporate A and U, not just G and C as before, in more in a better in more efficient way. So Jacques Zostak published several papers that he showed that yes, indeed, there might be non-enzymatic replication process which is with much higher fidelity and incorporation of all of the 4 nucleotides. Therefore, you can come to the point eventually to the moment where you have self-replication ribozyme. Now, according to the RNA world hypothesis, the moment of Darwinian evolution, and that's why I started to explain everything here, the moment of Darwinian evolution is only the moment with self-replication. So self-replication, once it started, you have already Darwinian evolution. And actually that fit whatever I showed you, information and structure. Simply, information and structure are the same polymer, the same type of polymer, but it fit very well the description I showed you. The problem here is that even if you assume somehow that self-replication ribozyme naturally will occur, how you can explain the polymer transition from self-replication ribozyme to the process where you have translation as a process, so the peptides. And that makes the entire process from short peptide, from short RNA through self-replication ribozyme. And after that, the transition, the polymer transition from self-replication ribozyme to translational process, absolutely so much complicated that I think it's a bogus. This is absolutely wrong hypothesis. Everything is wrong in this hypothesis. Not everything, but all of that step never occurred. In fact, in the Mother Nature, right now, there is no such a thing as a self-replication, meaning in a level of RNA, in a level of molecule, not in a level of entire cell. There is no such a process of self-replication, either ribozyme or anything that is known. I personally come from my laboratory experiments and everything that the field that I was studying so far is actually DNA replication. I consider myself a DNA replication guy and cell cycle. So my point is, according to me and according to many other scientists, self-replication never occurred, never happened, and RNA world is a wrong hypothesis. But the problem is that this is a most dominant hypothesis right now in the field. And many scientists continue to say it and to say, yes, we have to find somehow how self-replication occurred. And this is continue and continue and continue. And because to me this is a wrong hypothesis, we continue to bump in the same wrong hypothesis over and over and over again. So, and that's a problem, because science, sometimes it's not too much genuine, sometimes it's about fashion. You have fashion style of science. Sometimes something which is promote more papers, sometimes that it's more publishable, that goes forward. Unfortunately, that happened, but also in a long term, now we are speaking in a long term, more than 10 years, science is also self-correcting process, meaning eventually RNA world hypothesis will go where it belongs, to the history, and that's it. And RNA peptide world, which is the hypothesis which I am right now supporting, I think it's more explanatory. You have more explanation power. This is very important. How hypothesis explains the things more, and there's no gaps. As long as you don't have much gaps or explainable gaps, you're fine. But if
so how do you go from that hypothesis, Dmiter, from that peptide RNA world to a fully functional modern cell translation where all the players are fully developed and in place? Is that not a huge trajectory?
No. You have, in RNA peptide world, you have generation, something which is occurred as a primitive translation. Primitive translation means that you have slightly more chance to have a specific amino acids aminoacylated to the proto-tRNA, which is have a specific sequence, like GC-rich sequence. That's it. Once you have that process, this is already primitive translation. Now, because of the hybridization dependent process, that goes in a very short peptides, as I explained. But now, what about the replication? And that's very important, because without replication, everything will be anyway somehow destroyed. So you remember, it's a supporting structure of the information, meaning you're supposed to have somehow process of replication. Replication, yes, but not self-replication. So how you can explain replication in that case? And I can explain it very easily, because in RNA peptide world, the problem with the replication, not self-replication, the problem with this replication is magnitudes much easier to explain, because every single polymerase that we know of, nucleic acid polymerase, either RNA or DNA polymerase, actually the catalytic center is 2 or 3 magnesium ions. That is the catalytic center of any single polymerase that we know of. So a short peptide which somehow holds in a specific distance magnesium or 2 valent ions could be serving as a primitive form of replication and support the very short oligonucleotides that is already in existence and preserved. So you have very good explanation why you can have very early replication, although it's not super high fidelity. You don't need to be super high fidelity, because remember, you're in a situation of already Darwinian evolution, where everything that doesn't fit, doesn't go well, it's destroyed, but everything that goes well will continue to exist.
I still see a huge gap, Dmiter, because we know how complex a ribosome is. It's one of the most complex proteins, whole enzymes that we know of. It has about 80 proteins and ribozymes, and they say it is basically a factory inside of the cell factory. And there are so many things that must work together, and it has even internal signaling, and it has repair mechanisms, and all this has to be made by different enzymes and proteins and chaperones and cofactors. That's enormously complex. We don't even know exactly how all that works, and I simply disagree that it has been explained how the trajectory from your hypothesis to a fully fledged ribosome, how that can be explained. I don't see that trajectory explained.
Yeah, you don't see, because you're always working with the wrong model. And what I'm trying to say, you always bring the complexity argument again. However, let me finish. So can I finish?
Yeah.
If you go, there is a paper from Anoles and Anoles, and they try to look how the ribosome actually evolved, in what sections evolved. And they actually can dissect the ribosome in the early and later sections, evolutionary. Also, Gary Noer. Do you know Gary Noer? You don't know it probably. He was one of the ribosome gurus in the 80s. Gary Noer, almost, I don't know, he received Nobel Prize or not, I'm mistaken probably. But he showed that if you stripped out all proteins from the ribosome still will be functional. It will be low level, at low fidelity, but still will be functional. Also, it's shown very clearly that the actual enzyme of the ribosome, which makes the peptide bond, is the so-called peptidyl transferase center, PTC, which is in the larger subunit of the ribosome. And this is in the center of this larger subunit. Only this center can work and function as a peptide transfer. Everything that is doing this peptidyl transferase center is just making a trap trap where simultaneously makes the transfer of one amino acid, which is over the
amino acid tRNA.
Of the tRNA, it's a transfer to the other. So
tRNA.
this is a situation where you don't need even energy in that moment. You just have a small trap, kinetic trap, with the situation, and that could be functional without even having the entire assembled ribosome. Not to mention that the interaction is entirely based, so the tRNA, mRNA interactions, which is happen to be around the area helix 44 in a small subunit. This is where the AP center of the ribosome is. This is entirely based on a completely pure natural processes like nucleotide base pairs, Watson-Crick base pairs. And by the way, you don't need this to have. You can have this is this is completely natural process that you can assemble and you can use it outside of the ribosome. Meaning this is not a unique process for the ribosome by itself. So all of these interactions shows that you may have much simpler ribosome to function. In fact, the hybridization dependent peptide formation, which I proposed in the hypothesis that I'm presenting, shows the possibility that just because you have hybridization process, you can have peptide bonds. Now, I'm not talking for the efficiency here. The ribosome is super highly efficient enzyme. This is ribozyme. But this is due to many upgrades. The upgrades that the ribosome is done evolutionary. That doesn't mean it cannot function in much simpler level. Also, you can see how fitted, very well fitted is the tRNA with the pocket there. So the process of recognition between the codon and anticodon is supported by so-called clutch mechanism of the small ribosome. So meaning the tRNA, it's coming, and once it fit all the 3 nucleotides, it's grab it and turn it out. Once it's turned it out, elongation factor TU release the tRNA, otherwise will not release it. Those process of elongation factor TU, plus there is a one loop there, and so on, all of that makes the process of tRNA, of translation, ribosomal fidelity, very high. It's very high fidelity. But that doesn't mean you must have those kind of fidelity in the very first primitive stage. So,
well, Dmiter, let me interrupt you on this. I have investigated how many error check and repair mechanisms that are involved in the translation process, and including the error checks of the amino acid tRNA synthetase and the ribosome. I have counted 11 different error check and repair mechanisms. That means that the translation process, it must be very precise in order to minimize the mutation rate of the process. So if we have a higher mutation rate, then that means that the protein will not be functional. So how do you explain that this kind of error check and repair in so many levels, in 11 different levels, could have evolved in a stepwise fashion if in the case of a higher mutation rate, the proteins produced are not functional?
It's very simple. You don't have such a complicated system initially. Once you have more simple system, you don't need all of these corrections.
Well, but if the system is
once
simple,
you go from a very simple system, you always can upgrade step by step. And if you see the tRNA, for example, you will see lots of modification, acylations, phosphorylations, and everything is occurred in order to be recognized from amino acyl synthetases better. So you have a fitter. Now imagine you have not so well high fidelity. So what will happen? If by chance you don't have, if you have a wrong incorporation, so if you have a wrong incorporation, you will have completely, so you may have simply not surviving of the particular.
Exactly.
So, but whatever it is successful will survive. And now if you have something that it's modified and fit very well, it will survive better. And that something could be just a small step up.
But could it be that it doesn't survive at all? Because if you don't have a minimal set of proteins which work together in a joint venture, nothing goes.
Minimal set? What does it mean, minimal set?
I give you an example. If you don't have an import channel for iron or molybdenum or sulfur, what would happen? You would not have the availability of these elements, atoms, to make the cores of many proteins that require iron sulfur cores in order to function.
I do not understand your example. Can you repeat it? I simply have difficulty to hear.
Okay. There are many proteins which require in the catalytic core iron sulfur or molybdenum cofactors.
Yeah.
Okay. So these have to be imported by the cell.
Who said that?
Well, we know there are many import channels, membrane proteins, which are specialized to import. They have affinity to import either iron, which has to be chelated, or sulfur, or molybdenum, or many other elements which are life essential. They are imported into the cell from the environment. Correct?
Because you have that cell now. But if you don't have this cell, if you have different cell, if you have much simpler cell before, you don't need, you can just grab whatever it's in an environment.
I disagree with you.
So that's what it's all about. So you might have much more simple system at the beginning, and then to continue to upgrade. Hi, Will.
Hey, I just wanted to say I disagree with you, but I appreciate you still. You know, I think you're a cool guy. I love your glasses and your headphones. You know, essentially, I was listening to you guys, and I thought that, you know, the best proof for God's existence is, you know, based upon our personal experiences. You know, for example, me, you know, a couple of years ago.
Yes, but we are not talking about personal experiences now. We are trying to elucidate the trajectory of a simple from a polypeptide world, which is Dmiter is proposing, to a trajectory to a fully fledged ribosome.
Yeah,
so
but I mean,
yes, but you're off topic now, so I will put you on backstage again. Okay?
What?
Okay, let's continue.
So we're talking about something that could be more simple and still to develop. Now, the idea was that you need to have a specific enzyme to import a specific ions in order enzyme to work.
Yeah, you know, for example, to have nitrogenase, you need molybdenum, iron, sulfur. All that has to be imported, and then to make the iron, the cofactor, it is a very complex trajectory process. And many life essential proteins, they have these iron sulfur cores.
Yeah, that's not a problem. I mean, to us as evolutionary biologists, that's not a problem. If you, for example, do you know Lupus? The Lupus, he is a German scientist, which he described from. I mean, everybody know it, but he was kind of a very good of description how the very early motifs of the proteins could occur, and he described it's a called motif alpha. I don't know exactly how was the paper, but he described the very first motifs of the short peptide. So we do not talk for fully developed protein until the RNA peptide is finished. First, you don't have protein at all. You have a very short peptides. And environment is leaky. You have very leaky, although it could be separated because of the lipid layers, because of the small cavity, because of the small vesicles that you can have, because of adhesion processes and so on, because of sedimentary layers and so on. You may have a specific environment and compartmentalization, but that doesn't mean you don't have a fully permeable or leaky environment as well. So exchange of everything like organic moleculars, ions, and everything is very free to go anywhere. Now so what is our model? It's totally different. So you start with something which is entirely dependable from the environment and the surrounding. And once you have, for example, a Darwinian evolution to kick out and to start, this Darwinian evolution depends on everything that it's surrounding. It's not like an independence. And now developing of the metabolism and developing of all of this process of synthesis of your own things, it's a process which leads to some sort of independence, but that goes to step by step from very small, completely first dependent from the environment to step by step independence from the environment. And now, what about the evolution of the proteins? Initially, we're talking only for short peptides. The peptide works in a way as it is, for example, the actual amino acid composition. So if the peptides possess certain and certain amino acid, it behaves in this way. Meaning, for example, if you have more positively charged amino acid, it will interact more with negatively charged surface, or vice versa. Now, once this peptide became slightly longer or more longer, now we are talking for 20, 30, or 50 amino acids, you have a process of folding and formation of motifs. These motifs now depends on how actually the distant amino acid interacts each other and forms short motifs. Again, these short motifs are not full peptide, but they are just motifs which have a very slightly more complicated function. You do not expect to have a fully developed protein. Now, once you have more longer and longer peptide, now you have a folded motifs, meaning a 3D structure of the peptide bond, which create a situation of formation of catalytic centers. Once you have these catalytic centers, we can talk about proteins, because this one, in order to be synthesized, need to have a specific start and specific end codon. So to be translated in frame, but to be translated in frame, you're supposed to have certain system to recognize the start and the stop codon. But that doesn't mean it happened immediately. It happened with the entire process of evolution of the proteins from short peptide through short motifs, and after that longer motifs, and after that several motifs together, and after that fully developed proteins. At that moment, RNA peptide world is finished and started the real protein world that we know it. So that's the model which we are using to there is no any controversial situation where you're supposed to have a specific fully developed protein that will bring the specific ions in order to start and to capture the particular catalytic center. In fact, one of the oldest proteins, like for example, RNA polymerase, doesn't have any ion to function initially. So
no, that one maybe doesn't.
Whatever it's happening as a catalytic, this is adaptive function,
Yes,
which happened later when you have catalytic center to do it.
well, I agree with you, Dmiter, that there are many proteins which do not use metallic cofactors to function, but there are also many which actually do so. And of course, how that composition came to be is an open question. I've not seen a science paper which provides a compelling
I
narrative.
just told you. I just explained you the scientific way of doing it.
Yes, David, you did. I agree with you. You gave your narrative, and I agree as well that you do not have to provide all the details exactly how it could have happened in the minutiae of each single step. But nonetheless, I think it is there are very, very big open questions of the trajectory of having it starting from your model, from your hypothesis, to the complexity which is all are actually seen of a minimal functional cell. And for example, the membrane proteins which do the import job of metals, they have only a very short a sequence of amino acids which have affinity with the metal. And if that specific sequence is not there, they lose their functionality and their ability to recognize the specific metal that they are supposed to import. So that sequence has to be there. So we are going up to a point of atomic precision required to have functionality of import proteins, for example. And of course, you need to explain how did that configuration get there? And that's not an easy task to explain by natural means without invoking intelligence and intelligent direction.
Well, you always forget in entire all of that, whatever you said, you always forget the process of selection.
Now, I would like to ask you a different question, Dimitar. Would you agree that there are two possible, in a general way, in a general sense, two possible explanations of the origin of life? One is that it happened by natural means, as are your hypotheses, and the other would be the one which I propose, which we as intelligent design theorists and creationists propose, that an intelligence was involved to set up the whole process. Okay? So these are the two models, the two possibilities.
Okay, I can answer your question.
At what would you think would be expected? What would you expect to see in regards of the evidence or what we actually see in the molecular world in order that you would grant and say, oh, okay, I am seeing this evidence, this proof, that's what we see here. Hey, but that seems that it is better explained by intelligence rather by evolutionary manners. What do you think would that be? What would convince you to actually explain it through intelligence?
It's very simple. As long as we are able to explain the origin of life and evolution and all the steps in entire natural way, we do not need intelligent designer to do that.
Mm-hmm.
So,
but probably
what is the more okay? What is according to the scientists? What has more explanatory power So let me assume that suddenly it's appeared full developed cell out of the blue.
Okay.
Let's assume that you see that it happened just out of the blue. It's like in the magic movies. Shlap, and it's appear.
Yeah, but I don't think this is the
Now,
right approach or burden of proof.
in that case, if you take these particular appeared cells, completely appeared out of blue, nothing, and you start to study these cells, you start to see as well that all the proteins have some sort of relation. If you can very easily, I mean easily, relatively speaking easily, but you can draw a phylogenetic tree of any protein that we know of. Phylogenetic tree mean you have a motifs which is paired to the other proteins. The same motif, the same sequence you can see. That's why we have a protein families. You have a specific, the same type of motifs that can be amplified in any, in a different proteins. So everything as evidence that we have, and this is observation evidence now, observation shows that everything is built from the very simple blocks to make it bigger, to make it higher, to make it more complicated. So how you can explain, for example, the possibility, our possibility to draw a phylogenetic tree of any single protein that we can see? How you can do that? How you
Well,
can explain?
first of all, Dmiter, in order to have functionality of a protein, it has to have a minimal size. Like, for example, helicase has about 1,000 amino acids. You won't get the functionality of a helicase if you have 20 amino acids. You need a minimal structure and a minimal size of that protein. So,
Well,
taking
do you
just
need,
comparison,
do you need all the time?
Hmm?
Do you need all the time to have a helicase activity?
Well, of course, if you have just a random assembly of a random sequence, you won't have the functionality, and that's what we
So
want.
I can tell you because I am a replication guy. I study replication many years. So you need helicase activity if you have a relatively long DNA, for example. If you don't have relatively long DNA, you don't need helicase activity. And in fact, the helicase activity is not too complicated as a process. It just to separate two strands of the DNA, and that can happen, and actually has been developed in the history of life. Very different helicase activity and different functions, meaning they are different types of proteins. One feature could be developed independently several times.
Well,
So
this is my point, Dimitar. In order to have whatever functionality that you assign to a helicase, it has to have a minimal size.
of course,
Okay, so how do you get to that minimal size if any smaller size will not provide any function?
with minimal needs, because you have minimal needs. If you have a minimal needs, you have minimal functions. You have minimal size. If you have bigger needs, you have bigger functions. For example, the very first stage of LUCA, it's called proto-LUCA 1. This first stage of LUCA have only a few peptides, should have only a few peptides, and several ribozymes. The peptides have only a few functions. One is amino acylation process, and the second is the replication process. nothing else. And probably some catalytic function of transition of some simple chemical reaction, maybe that facilitate as the beginning of the metabolism. But you don't need anything else. At that environment, if you put in this environment highly developed helicase, it won't function because there is no need for that at that moment. At this particular moment, you don't need that.
Isn't that the point which I'm trying to make? That in order to have a function, you need, in many cases, many proteins working in a joint venture together. And if they don't work together by themselves, they have no function. So why would natural occurrences come up with a protein which by itself has no function?
But that's what I'm trying to explain to you. You don't need, so the needs always come, so you have a simple system, simple needs. You have slightly more complicated, and you have slightly more complicated needs. It's all about adaptive function. So, for example, let me duplicate. So, if you have, for example, you have a very simple replication and not enzyme, but very simple replication motif that holds, let me say, it holds 2 magnesium ions, and that perform a function of low fidelity replication. And now, because the system is very simple, just with few peptides and few relatively short RNA, at the moment when you have additional slightly, for example, duplication or joint or mutation of something which is come there, and it happened to be with slightly higher fidelity at that moment, you have slightly better performance, slightly better adaptive function. And therefore, such an organism will survive better. Do you understand?
But are you not snaking into your explanation always a teleological component where you say, oh, it will not, it has to survive or it will have function, but at that stage, molecules, they do not have any need or aim to survive or to function or whatever in that sense.
Okay.
We are just laying around.
If you take a stone and throw it in the water in the river, do you think this stone actually have some needs to survive?
Say that again, please.
Well, it's stone, right?
So a stone isn't alive, so it doesn't have to survive.
Doesn't matter. Just asking, is it need this stone to survive?
No.
No. So if you have complicated system, slightly more complicated, which is life, what could happen with this system?
We are talking now about your hypothesis, which are very simple peptides joined together, amino acids with RNA and so forth. And I think at that stage you cannot say that is something that is alive. So these are just molecules. They do not have any aim or urge or need.
That's correct.
Rectify.
They're stones, huh?
There is that doesn't mean there's no need to survive. They just survive.
Right, which refutes the argument.
Well,
you don't
have, of course, implies
you don't need have to have. You don't need
that stage.
Yeah, exactly. But you don't need to have some willing to survive. It's just the system by itself. It's more stable than the other. That's it. There's nothing different. You don't have will to survive. You have more stable system. If you have, for example, peptide, short peptide versus slightly longer, let me say, put 2 amino acids over this short peptide, and that makes slightly higher fidelity of replication. this system will be just simply more stable and will survive and will give the next generation. That's it. It's so simple. You cannot talk for moleculars that have some willing to survive.
Gentlemen.
Yep.
What's going on?
Hi. I'm sorry. I've been wanting to ask you some questions, Dmitry, for quite several minutes.
Yeah, I just want to interrupt you. I have put Tony and Leo in timeout because both are name calling each other. I have said several times I don't want to have personal attacks at my channel, so please stop it, or I just want to not have you anymore at all at my channel. So that's a final warning. Okay, go ahead.
Hi. Okay. Yeah, I wanted to first of all mention that I've got more of an inorganic chemistry background myself. As a nanotechnologist, doing microchip and MEM R&D for several years. I've always had a fascinating interest in the organic chemistry side. So I've got a lot of questions, particularly about some of these claims about RNA. People in chat have been mentioning you've got a paper out there that details it. I've been wanting to ask more of these questions because I'm actually more on Otangelo's side. I hope I'm pronouncing your name correctly, sir.
Otangelo.
Otangelo, thank you.
Okay.
Where, despite the fact that there appears to be massive complexity within what the director for the Human Genome Project, for example, labels DNA as the language of God. Just throw it out there. It's not so much that it's just that it's a complex system. It's that it's universal, and that there is an extreme amount of effort that goes to keeping it universal. It goes to keeping it from not basically self-destructing. And as an inorganic chemist, which deals with a lot of peptides, particularly on the plastic side of things, the inorganic side of things, which has similarities with organic peptides, there is a huge, probably the 99% of the entire job that we do is trying to keep our complex reactions from simply, like I said, self-destructing. They don't naturally stabilize. In fact, it's quite the opposite. They naturally destabilize. And I think that we can see that example going into life as well, particularly with the fact that there's really only 5 nucleic acids to even begin to talk with. When for example, my first question was, when you were bringing magnesium into RNA chains, for example, how did that apply? It didn't seem to click in my mind. I'd like to hear more about that if I could.
So this is because so you missed the point for the Darwinian evolution moment. Mm-hmm. So this is the whole idea of Darwinian evolution, about the stability. Of course, if you put everything, if you put just in environment, whatever it is, molecular system or whatever it is, it will destroy. I mean, eventually you have conditions, harsh conditions, all kinds of conditions, the time, the process, chemical reactions, and so on, it will destroy.
Correct.
And that's true, that's fine. But the moment when you have Darwinian evolution, you have a system where you have a system of information which determine a specific structure. And at that moment, this structure harbor and could potentially or could stabilize the informational source. At that moment, the system, when it exists, this system, if it survive, have the ability to be more complicated, so to be more adaptive, because a change in the information could reflect immediately change in the structure. Once you have change in the structure, could be either negative or positive in terms of destruction or surviving or stability. So if the change of information happen to be more adaptive, just by chance more adaptive, it will survive to the next step, to the next generation, because there is information. My point is, you have a huge difference between the system, which is just open system, and the system which is still open but possess Darwinian evolution relationships.
Okay.
It's 2 different kinds of matter.
Well,
yeah.
Well, I'm also going to let you know, I'm also what they call an information scientist. I've got an MISM, and so I look at a lot of these things that we're talking about, these systems, as code, essentially.
Yeah.
But so you're basically assuming, it sounds like, then, that the code that's reliably replicating itself, is creating all the known life that we see, is just a good, for lack of a better word, a good random accident, and that all of the other the false bad accidents, the stones that didn't need to survive, as you were trying to analyze earlier, just disappear.
Immutable, can you continue? I'll be back just in 5 seconds. Too much coffee today. Sorry. I'll be back.
So I'm not sure if you're referring like directly towards chemical stability, if you're referring like to the process of replication itself.
I'm not sure if that is actually anything to do with what I was asking. I was talking about as far as what he was talking about, as far as the what I would call the assumption that random code is able to remain useful.
Yeah, as long as there's a selection course, yes.
So that wouldn't that, yeah. So natural selection is not necessarily true. In fact, I also
what do you mean by
Aristotle's physics premise from 350 BC, where he negates natural selection. So it seems like a more atheistic version of God, is to say, well, natural selection did it instead of God doing it.
What do you mean by it not being true necessarily?
Well, like I just said, it's not self-evident. So, and there requires to be evidence for something to be considered to be true or testable. Natural selection is not testable. It's just an assumption that something else except God.
Natural selection is a phenomenon that's present in statistical environments. You can test that in chemical species. You can test that on biological species.
No, you can't.
Yeah, you can.
Now, how do you? Okay, so you're giving agency to something that you say you can that doesn't have agency. That's what I'm saying. A lot of this is wrapped around assumption. I want the actual scientific evidence.
Right.
Again, so we've got good philosophical background to show that natural selection is not true.
So, if you were talking about a basic chemical reaction, right?
Right.
That chemical reaction is going to be determined based upon the equilibrium constant is going to be based upon
the laws of chemistry, right?
Yeah, exactly.
Which is nothing to do with natural selection.
Well, yes, and part of that, the reason natural selection is able to operate on that is due to the chemical stability of both the reactants and the products.
Yeah, no offense, but that sounds like a purely religious statement to me, though. It's just assuming.
How exactly?
Like I said, it's just assuming. It's the same exact logical statement as saying God did it. God is natural selection.
How exactly? Because
again, as an information scientist, so I study chaos theory. I'd study information systems. Part of my professional career is taking a huge amount of disparate amount of information that has nothing to do with anything among itself, and help create automatic and anatomical filters to understand and basically disassociate all the disparity to make it useful for users, for the end user, for the consumer. Right. And so that doesn't happen naturally. It can never happen naturally. Chaos theory, one of the very basic premises for that is that the more stable a system becomes, the faster it breaks down. And we see that today, everywhere in information science. In fact, it's getting so complicated, so impossible to try to regulate just the human side of information, much less this massive amount of universal information in cellular structures, that we're starting the next quote unquote paradigm breakthrough for information science is relying on Elon Musk's Neuralink. We're basically hoping to directly plug our own brains into our computer systems to be able to process this stuff faster. That's how crazy it is. And it's nothing compared to the super complexity and the uniformity within DNA code. So to say that DNA is stable just because someone said it had to be is pure religion.
No, DNA is stable for a variety of reasons. Its structure is one of the inherent properties that makes it do that.
So that's what doesn't make sense to me. So again, as an inorganic, I look at the 64 codon groups, right? Okay. We got the 4 nucleotides that based on the 4 nucleic bases that create the 64 possible codons. So that right there, that can be a huge mess by itself. Again, I'm a man.
So if we're asking, hold on. So if we're asking about the stability of DNA,
you're not allowing me to make a point.
I know you wanted to talk about the stability of DNA and RNA, which
I was making a point why it's not possible, and you're interrupting me.
Go ahead. I didn't know you were trying to reach that point.
Yes, yeah, I'm trying to stay on topic. So there's a huge variability in all genomes. They use all 64 of these codons, which is the total possible amount of codons. So the chance for catastrophe is there. It doesn't have to be stable, in other words. But the fact, for example, that all DNA, all genomics, in all plants, all animals, all microbes, all life uses the same exact 3 start-stop codons. That is not random. That is specifically non-random. And so for the idea that it could be random and that we should see examples of it just does not exist. There's a lack of instability, like you say. That's your argument. It's just stable just because. Well, it's not just because. It's because it's been specifically written that way. Where's the examples of where the other 3 the other codons, for example,
so
as a start-stop codon besides the 3 that are used universally? Where are those examples? Which is what we should see in a truly random, quote-unquote, natural selection type environment.
Not necessarily, because one thing you could say is that the stability of the relationship between codons have something to do with how amino acids interact with dinucleotide or trinucleotide chains.
I can't see that because all of this, what we're talking about, the DNA and the RNA sequences, all of that predates the amino acid protein chains. That's all external to the nucleus. That has nothing to do with DNA.
Right. Although I don't see why that precludes there being an interaction to explain.
It's literally, I apologize, it's literally worse than the chicken before the egg. Nucleotides have nothing to do with amino acids other than just activating, like I was saying in text, activating the pre-existing or quote-unquote preordained, so to say, other external components outside of the nucleus. The information is completely segregated and independent of the rest of the biological life cycles. As their DNA activates the protein production chains, the protein production chains never do anything to touch DNA. If they did, it would just further destroy things.
Right. And
okay. So we're going in a deep.
Well, I try to keep it simple. It's one-way information. So and that's the other thing. RNA is a packetized information chain. It's a one-directional thing.
Let me try to go a little bit back. I understand your point, but the reality is slightly different. So, how you prefer to call you sfbmod?
I don't know. sfbmod. I apologize.
Just call mod for short.
Sure. Yeah, I don't do a lot of these online chats, so I've not come up with a persona yet. I apologize.
Okay, so sfbmod. First, do you acknowledge that there is mass extinctions in the animal during the of time?
Yeah, that's basic fossil record
What
stuff.
do you think? What do you think? Yeah, what do you think happened in mass extinctions?
Things die, but I don't understand what that's got to do with the.
I'll come to this point. Mass extinction creates a situation, environmental situation, where simply the biological system of this particular species does not survive.
Okay, so
meaning that there is a conditions where the biological systems are not able to handle it, and they die. It's so that, however, about the stability, that means that by saying that if it's too complicated system, it's more fragile. It's not.
Yeah, that's chaos theory.
It's not exactly accurate because
that's basic chaos theory. Yeah.
It is not exactly accurate. It is stable and relatively stable in this particular environment. Of course, there is limitations. There is a limitations where the system cannot survive. So it's not to say that if it have more complicated, it will be more fragile. No, it will be more adapted to the particular environment.
Right. So first of all, when you're talking about mass extinctions, from my understanding, all those are external to life. Life doesn't self-exterminate itself. If we're talking about the stability versus instability of the code of life. If the life code became unstable, the thing just doesn't grow. It doesn't even get born. It doesn't even start in an embryo. It just dies as a random mess of cells. Yeah. So it can't. There's no mass extinctions would not explain the infallibility of mistakes in code for life.
My point was that there is some limitations which the actual biological system, even that it's very complicated, could stand for. In fact, as simple as the system, which is true, as simple as the system is, as more stable will be on a harsh environment.
Yes.
And that is
That's
very,
yes, right.
all of extremophiles are bacteria, which is much more simple in many ways.
Not at all. No, there's bacteria genomes that are more complex than the human genome, but
No, it's
regardless,
not more.
the
No,
thing is that they're still using the same exact information codon set as all life does. It's not just that it's got more complex or more simple.
Majority of bacterial
It's
life.
universally used.
There are some that are exceptions, though.
So
Well, the point that's just not a valid point in my mind.
So it is what it's to say about the complicated systems. They are not just complicated, more complicated because are more
agree.
stable. They are
Right.
more stable because are more adapted to particular environment. That's why.
That's not necessarily true.
Yeah. So
If we were to take one of us or both of us together to go live out in the Arctic without any clothes or technology, we'd die. We'd be out of our environment. So I just that argument
that's
doesn't.
what it is all about. Yeah. So
Well,
about the stability of the RNA and about the stability of DNA. I'm working maybe 20 years in the DNA replication, DNA repair, and the cell cycle.
Mhm.
You have constant process of repair. If I show you just 1 second, I'll show you back Otangelo. Probably you will start to have a headache of this picture now. But
Actually, I would love to have that conversation with you. I'm very familiar with line 1 processes, transposons. I'm very interested in the histone data
so
archival.
let me show you, or let me show you again. Let me show you again this particular picture, and it will continue from that point again.
So
Well,
the record is straight. I have a
changing subjects.
master's in biochemistry. I do know this topic.
So here is the definition of life and Darwinian evolution. When you have a system where you have informational source which determine the function or structure, and that function and structure actually maintain somehow the informational source, and that is the process. Just want to scroll a little bit down because it's okay.
Yeah, it doesn't work in information science.
So
It's the exact opposite is true.
and this is so simple. I'm not saying that there is a reason to be so simple because we are looking for origin of life. That's why.
Right.
But by specific rule, which is happen to be there is 2 rules known for now. For that one, it's called genetic code, and another is base pairs, Watson-Crick base pairs. By specific rules, the information can determine or execute as function or structure, meaning, for example, proteins or peptides.
Right. I was talking about that earlier too, right?
Which maintain the information. Once you have this system, this is how the Darwinian evolution works. Now, if you put this system in environment, that system might perish, of course, might be destroyed. But if it survived, the information can execute the same type of structure here and over again. Now, if you have mutation in the information, something change in the information, that blindly will go in the structure and function. And again, it's a subject of environmental test. So either will survive or not. And this is how the system works as a Darwinian evolution.
Yeah.
Now, there's something which, as a description, as a definition, I should show you. So you see the definition of Darwinian evolution, which is absolutely identical for life by itself. Darwinian evolution is a dynamically stable system. Not just stable system, dynamically stable system where information determines its own supporting structure. So that is what the Darwinian evolution and life it is. It's all about supporting dynamically stable system. So when you're talking about stability here, you don't have a situation where just more stable, like for example, crystal. One crystal is very stable, but it's not dynamically stable. It's a steady state stable.
So it doesn't explain origin, though, does it?
So this is the definition of Darwinian evolution and life per se. There's nothing to do with how complicated is this. It could be very simple.
Right. No, I agree. It's not about the complexity. The proof of God in DNA is it's universal, not that it's complex. But I've heard Darwinists say the exact opposite of what you defined it as, that it is unstable, and that's why we get fish to dinosaurs to birds. The fact that it does change randomly is how that unstable stability is how we get evolution of species.
Yeah, that is what my point is. It's a stable, but it's a dynamically stable. For example, the DNA, it's not a magic molecular. The DNA is subject for modifications, and we see that in our laboratory and our experiments.
Right, that's what I want to talk about.
The DNA could happen with the DNA pretty much everything that you can think of, either to be cut, deleted, amplified, could be modified, to be changed. Everything is happening.
Archive retrieval system. Are you familiar with the histone and the epigenetic marker type?
Yes.
I'm familiar, of course, as a molecular biologist. It's my subject, although this is not exact my area of study. Now, the epigenetic is just epigenetic, meaning level above the genetic.
Right, outside of genetics, right.
Yeah, but it's still information, it's still coded. So it's still, you have a code.
It's still regulated, right.
Which is about the, could be phosphorylation, could be acylation of the histones, could be methylation of the histones. All of that could happen. And they could happen in specific histone, in specific pattern. And that could be read, you can read that from different, from the different enzymes that can recognize this particular pattern.
Correct. See, that's so fascinating, in particular about your research. You were saying you study the repair mechanisms as well. The retrotransposons, for example. So again, as a coder, as an artificial intelligence coder, I look at that. I see those are examples of actual cognizant artificial intelligence subroutines written into DNA. They go in there and they make sure that those histone changes and those epigenetic markers, those tags are put in the right place, that they're put in there in the right.
Are you talking about
at the right time? Because if they're not, it will damage things.
Do you see?
Are you talking about transposons?
There's actually more than just the retrotransposon. That's just the more glaring example, I think, of what I would argue is definitively an actual AI program inside DNA.
I mean, most times you're referring to like promoters or enhancers, not transposons itself. Transposons have a tendency to copy and paste themselves more or less randomly into other parts of
the Yeah, that's the thing. There's a bunch of papers out there for the last 20 years now talking about how none of this is random, and there's been no junk DNA now. We've got basically, that's how I talked about it earlier, a back-end type archive data transfer system that archives.
So if you're referring to the ENCODE paper,
makes it active temporarily, such as like liver cells and brain cells do. When the liver finds something like a new poison, new toxin comes into the system, there's archived pre-engineered data inside the DNA for the liver cell. I mean, it's in all cells of the organism, but the liver cell specifically says, okay, let's unarchive this particular piece of histone encased, or whatever you call it, organized code to create the proteins we need to attack this poison. And when it's done, it packs itself back up. It's exactly like back-end to front-end type like code data, like we do here, like YouTube uses. YouTube is a back-end, front-end server type service. And then there's all kinds of checks and balances with it. And that's what I'm talking about. That not only is there no randomility into it, there is all kinds of cognizant AI routines in DNA, making sure that if something does randomly happen, to try to fix it so it doesn't cause disease or like Dmiter is talking about, death.
Yeah, but how?
It's somewhat.
How this proof intelligent designer? I don't understand.
Well, because again, as an information scientist, like I said earlier, I don't know if you caught it, if this stuff could happen kind of on its own, we could create a stable system that could just kind of keep itself stable. As the assumption, nobody in the information big data world would have a job.
Let me ask
we did that by now, and we all be going home eating Pop-Tarts.
For example, do you see the entire process?
I try to, yeah. Again, that's what I'm saying. There's no resemblance of junk DNA any longer. Not only do we know it's all archive data, but it's all three-dimensional matrix.
It's
all pre-engineered. It can only break. It can't seem to fix the.
It's not pre-engineered. It's not.
It is pre. It has to be. Whenever
No.
I see archived code data, that is pre-engineering. When I see multi-level, multi-dimensional, when I see information on top of information, there's a dual information code. So when you start at one sequence, at one codon, and you read it, it creates a certain what do you call it? The messenger RNA thing to create a protein stream. You start at the sequence right after that, it will create an entirely new useful protein. That's something that can't just be random.
Yes, specific regions of DNA will do that. Although there's a lot of other DNA that exists, for instance, short tandem repeat and intronic elements in DNA that don't have any function.
Well, that's just it. That's the same thing in my code. I've got all kinds of code that I'll either have you want to call it in the DNA code inactive genes that will still be something that like a polymorphic function call for whenever I might need it later. It's the same exact concept. We don't
Right.
And then you use the word polymorphism in genetic code. That's not random.
Right, except for the majority of intronic elements within genes do not ever have function.
That's not true anymore. Again, what we used to
What
assume
paper are you referencing?
junk DNA is either it's not junk, it's archived code data for specific things, or it's scaffolding.
Are you referring to ENCODE?
I don't know what that is, so no.
So ENCODE was a project commenced.
Oh, ENCODE. Yes, sir. No,
Yeah.
I'm not referring to that. I'm talking about DNA.
Right, and you're saying that the majority, if not all, DNA has activity.
Basically, again, what we used to assume was just junk DNA is either quote unquote inactive DNA, which means that it's still useful code, just temporarily turned off, such as the example I gave with the liver cells.
Right.
Or it's actual physical scaffolding, because the DNA is also, it's not just a one-line code. It's the shape of the DNA, the changing of the shape of the DNA over time affects how the code is translocated, how it does things.
Right.
So it's not just where the gene is in the sequence, it's where the genes are physically in three-dimensional space. So there's code within DNA that basically forces certain genes to align properly as well in a three-dimensional matrix, which again,
so the point of epigenetics, though, is that information is dependent upon the proteins acting on DNA.
No.
DNA.
Proteins never act on DNA.
Huh?
How does a protein act on DNA?
What do you think a histone is?
Well, it's again just
you're talking about histones now. Certain modifications to histones will cause either repression or an uptick in protein activity.
right.
Although, do you not think histones are made of proteins?
Well, that's just the thing. The histone just triggers where and when the what they call it, the little circular, the pattern it makes, the little coils it makes, but it doesn't change the DNA. The DNA never changes.
Well,
Yeah,
I don't understand. I completely don't understand your point. Meaning, first of all, yes, there is epigenetics. That's true. Yes, there is a modification of either histones, or there is a modification of DNA as well. You can methylate. There is a CPG islands, for example, which happen to be next to the promoter regions of almost 95% of the genes in the eukaryotic genome. And now what? You have a specific enzymes, methylases, which either add methyl or remove the methyl groups from the CPG islands. And based on that, they can
the DNA around, right?
how active will be the actual gene. But the problem for what I'm trying to say, although this is correct, I don't see intelligent designer here. I don't see why should be intelligent designer here.
Well, again, as a coder, it doesn't seem possible to be random.
No,
so
it's not random. But you
don't want to see in design. They just say, well, natural selection must have done it.
Yeah, but you have to make a difference between just something that it's not random and have a specific pattern. Doesn't mean automatically this is not equal to intelligent designer.
So again, if it has a purpose, if it's not random, then it's engineered.
Okay, so let's ask
one question of. Well, it is selected, not engineered. The right term.
No, it's engineered. Selection is irrelevant. See, again, I'm an engineer and I'm a scientist. I've made microchips. I've done rocket science. I've done all this stuff, and so it's never selected by itself. We have to engineer these things. When I look at I look at the code for life, and I look at the universal ability for code for the life code to be able to fix itself, as you said, you also study the repair mechanisms. That's not random. That's the exact opposite of random. Again, applying chaos theory principles, if this stuff were random, then it would be self-destructible.
I agree.
DNA would exist if it was random.
See, the random and not random. If something is not random and have a specific pattern, that doesn't mean automatically that it is ID created. That means it is selected
Fine.
to function as well. Well,
I can agree with that, but that's a religious statement. That's all assumption.
So how is that a religious statement exactly?
Because when you see design, you again, as everybody from C.S. Lewis to Robert Jastrow will say, that denotes a designer. But regardless, that could also be made as an assumption. The fact that the lack of assumption is that it's not random. There is no randomility. Either God did it or natural selection did it. Seems like the only current conclusions.
Okay, and even if I were to grant you the idea of how God did it, when I ask you how God does it, the mechanism you're probably going to put forth is due to chemistry. Once again,
I would state something like Sweebach, software engineering.
So chemicals,
I do when I write code.
So chemicals are forming in such a way that it doesn't require the forming or breaking of nucleovalent bonds?
Say that one more time, please.
I don't understand if an intelligent designer is acting on the system to create a desired result, how this mechanism is being put forth without already natural mechanisms existing.
But it does have natural mechanisms existing already. What do you mean?
You have
so that's the point. If there are natural mechanisms which already exist, why
they exist on their own? So again, proteins cannot create nucleotides. They're entirely different chemical sets. As a matter of fact, and I do need to go. I apologize. I need to go take my son to work real quick, guys. But when I get back, something that reminds me, Immutable, and I really appreciate you saying it that way. The question that I want to ask, I want to leave with you guys, and I want to see if you can give me an answer. I've been asking this question for 20 years now, ever since I was back when I was doing my chemical engineering degree. Was about nucleotides, about phosphates, specifically the phosphate bonding. How does a nucleotide form in a free-floating phosphate bath? Because I asked my professors that back then when they were teaching all this. When we did the experiments, if you put nucleic acids, if you separate them, whatever, you get them out of the genome stuff, and you put it into just a random phosphate bath, they instantly react into phosphates. The phosphate bonding creates polyphosphates. So let me get back to you, but that's the answer. That's one of the answers I wanted to try to get to. Again, I've been asking for 2 decades. How does a nucleotide form in a natural phosphate bath without the pre-existing, super complex nanotechnology factory of a living cell to keep them separated?
Well, the process is very well studied. It's called polymerization. So you have nucleotides, which based on a template you add to the actual nucleotides. So
how does it work without the nucleotides?
So
how do they when they hit a phosphate?
The answer.
I think what you might be asking about, and correct me if I'm wrong, you're asking about nucleobase formation.
Basically nucleoacidic formation. The nucleobase goes into the acid, and then when they combine into the phosphate backbone chain, if the phosphates are not carefully restricted, they'll instantly react into polyphosphates. Is the problem?
Okay, so what you're asking about is the particular phosphodiester linkage in something like an RNA molecule.
Hold on, I couldn't understand you. Say it again.
You're asking about the particular phosphodiester linkage between 2 molecules of RNA.
Well, not necessarily RNA. That's an entirely different topic to me, but just
or
DNA.
Any set of
nucleotides in general. Yeah.
Yeah, any set of nucleotides is what you're asking about.
Yeah, but again, I need to run real quick.
No, that's fine.
I'll mute myself. But yes, basically, the problem that I'm hoping to get addressed with that is if you put acids in a free-floating phosphate bath, where there's no phosphate blockers, there's nothing else involved, then how do you keep them from reacting instantly into polyphosphates? I'll be right back.
What does it mean, polyphosphate, in that case? I don't understand his question very well.
A nucleic acid chain that's with multiple phosphates instead of a monophosphate like a nucleotide. BRB.
Well, part of that is an energetic barrier.
So, yes.
They'll react together if there's more than one phosphate nearby.
So you do not add to the. So when you do polymerization process, you do the polymerization process with the 3-phosphate. So you have nucleotide 3-phosphate. So let me explain. Submol. He's gone now, so I cannot explain to him.
Yeah. SFP, someone here, John Maddox, asked if you could leave your email. He would like to get in touch with you. Can you put it here in the private chat so I will forward it to him? Thank you. Yeah, go ahead, Dmiter.
So the polymerization process, it's a very kind of strange thing, meaning the phosphate bonds, once it's formed, the phosphate bonds, they're highly energetic, meaning you have, in order to make a phospho bond, you need a huge energy barrier, as Immutable said. So that's why it is happening by enzyme polymerase enzyme. And that's why in before the actual polymerization as a process to happen, the energy, this nucleotide is already activated, meaning you have a third phospho site which activates and makes, so the energy phospho bond goes, goes from this way, the energy barrier goes first up and after that goes down.
But that enzyme was not extant on earlier, Dmiter.
So you have 3 phosphate nucleotide, meaning the nucleotide have already added the energy necessary for the actual formation of the phosphate bond before going to the polymerization process. And the actual formation of nucleotide to nucleotide base pairs, they're not that strong. This is a hydrogen bond interactions, which are very weak, relatively weak. So they can fit each other, and once it fits, then the reaction of phosphate bond it happens. So and this is a process, the same enzyme doing it. It's like a palm. You can see it's like a palm and hand. So it goes. The actual enzyme goes like this. It takes the free nucleotide at the free nucleotide, and after that it's formed the actual phosphate bond. And this is very well known process. I don't understand where the problem is. So it is understandable, and it's studied very well, this process. It's my area. Yeah.
Well, you need the enzyme for making the polymerization, which was not there on the prebiotic Earth. And I mean, all these hypotheses, like the RNA on clay and so forth, these experiments have failed.
I just explained how many different hypotheses for origin of life have, and some of
which one works? Which one has worked?
Yeah, but this doesn't mean all of these hypotheses are correct.
Well, that's my question. Which of those hypotheses has worked actually? I don't know of any. Maybe I am not well informed, but I don't know of any of those proposals.
Yeah, the first hypothesis that it's could be more accurate than the other. It's the hypothesis which have less gaps in explanation. The better hypothesis is the better explanation power have, meaning could explain more and more things and doesn't have a huge gaps in explanation. Right now, so here it's again, I'm coming back to there is many hypotheses for origin of life. Unfortunately, this is a very specific area of science where you cannot go back in time and check it exactly how it happened, but you can show proof of principle in laboratory that this particular mechanism might work. Unfortunately, from the modern science and experimental biology at the moment, we don't have much progress on that. the only progress that we did so far is creating a bunch of ribozymes based on the RNA world hypothesis. And also there's something else I have to tell you outside of science, funding. Unfortunately, there is only few laboratories that actually work on the origin of life. One laboratory, of course, is in Harvard with Jack Szostak and several satellite laboratories in Harvard. I think Arizona State University with Sarah Walker. She also is working something with the origin of life. They in California. In California, probably there is one or two laboratories that might do some experiments for origin of life, and that's it in America. In Europe, there is probably in Norway there is one laboratory, and that's it. So in fact, there is several, just only few laboratories that actually
Japan too, right?
That perform experiments in origin of life. And the problem is not just there's no much laboratory. Many laboratories want to work in origin of life, but there's no funding for that. In fact, most of the funding goes for cancer research or something like that, and there's no money. So the funding for origin of life usually comes from either NASA or National Science Foundation. So, and that's a limited sources. And this is one of the reasons that we didn't do much progress on the origin of life research.
Well, I know there is a lab in Tokyo in Japan, and they got in 2010 a fund of $100 million to do abiogenesis research.
$100,000.
Now $100 million.
No, that's a different. I know what you're talking about. This is very interesting funding, but this is about proof. That's about proof that information can be created, meaning can be experimentally proven that could happen. And that's more like I think it's more like a bioinformatic proof than research. I don't know. And also the conditions of this particular grant is very strange. First of all, you have to apply for this grant, but after that there is only one will be selected to go further. And this is a very subjective process, which we don't know what will happen with this. Yeah, I mean, it is probable available source, but I have no idea how it will work.
I see. Yeah. Well, Dmiter, I will keep open the chat, but I will absent myself a little bit, so you guys will be free to keep talking to each other. If sfbmod comes back, John Maddox asked about his email. If he can leave it.
Let's finish for now, and that's it. I want to say something. I'm starting a new job, a new laboratory, which is dedicated to stem cell research, and that will be on October. So October, I will start actively to work. In that case, I will not be much available for chatting. So yeah. Whatever you can.
Set up some debate with Fazale Rana or someone else that wants to have a chat with you, a debate with you, engage with you while you are still available.
Oh yeah, I'll be available, but I'm probably after September I will no longer be too much available.
Okay, that's okay.
Okay, I can say bye for now.
Okay.
And, so that's it. So I'm open for questions always.
So thank you very much once again for being at my channel, Dmiter. It was a pleasure. And let's set up again. Whenever you want to come in, feel free. We will keep in touch through email, through Mike, and evolve our ideas and exchange information.
Well, it's still 11 September, so until the end of September we might chat again a few times. So it's okay.
Sure. Okay.
Okay.
Fine. Great.
Bye.
Thank you very much. Bye bye. Okay. So guys, I will stop this stream. Thank you, everyone.
You get going.
I'm sorry, Immutable?
Yeah, I'm also going to get going. It's about time for me to eat.
Yeah, for me too. So see you next time, guys. Thank you for being at my channel, and till next time. Bye bye.